G'day, my name's Dr. Jeremy Ray and welcome to my lecture on epigenetics. So here's our objectives. We're going to define epigenetics related to human health and diseases. We're going to review very briefly the structure of the human genome, chromatin and nucleosomes. We'll describe the major mechanisms involved in epigenetics, which include chromatin remodeling, histone remodeling and DNA methylation. We'll relate that to control of gene expression, and we'll give examples of various diseases and conditions that result from epigenetic changes. So here we have a good definition, I think, from the Encyclopedia of Toxicology. Epigenetics is a study of non-genotoxic, reversible, heritable mechanisms that influence gene expression without changing the DNA sequence. And here I've put an interesting quote about the old dispute between nature or nurture. And that's kind of very simply where epigenetics is at, is the debate about how much of your characteristics depending on your genes versus how much depends on the environment that you've been raised in. So here we have a useful overview diagram from the National Institutes of Health, which contain many of the key points that we're going to work through today. So a couple of important epigenetic mechanisms are DNA methylation and also modifications to histone proteins, and you recall that they are essential to the structural integrity of the human genome and chromatin. We can induce epigenetic modifications in a variety of ways. These include your age, diet, smoking, various chemicals, drugs and disease states. What we've seen is that we have altered epigenetic markers in diseases such as cancer and some developmental disorders, and increasingly we're seeing that these are able to be passed down through different generations. Therefore, if we understand how these epigenetic mechanisms modify gene expression, how nutrition interacts with this, then we can positively influence human health. The first mechanism we're going to talk about is DNA methylation. And a simple way to think about it, it's like a switch that turns on or off gene expression. When you add a methyl group to a certain position in DNA, it can turn off gene expression. We also call this silencing. It's one of the most well-studied epigenetic mechanisms, and the methyl groups are specifically attached to cytosine bases, which you find in the CPG islands of promoter sequences as illustrated here. Methylation also drives the formation of heterochromatin, which is a more dense form of chromatin, and that also reduces gene expression. As a result, you get stable long-term repression of gene expression. What we see in some cancers is hypermethylation of tumor suppressor genes and conversely hypermethylation of proto-oncogenes. This mechanism is also important in the following processes: tissue-specific gene regulation, genomic imprinting, which we'll talk about shortly, and X chromosome inactivation. So to understand the other mechanisms in epigenetics, we're going to very quickly review chromatin structure in histones. We did this previously, and you'll recall this diagram from before, reminding us that DNA is packaged in chromatin structures in our cells, and that it's composed of double-stranded DNA that is called around what we call histone proteins, and these form structures called nucleosomes. And the presence of these structures represses transcription of genes. And what we've seen is gene expression typically requires some type of remodeling in order to make these regions accessible to transcription factors in RNA polymerase, and relief of repression is caused by changes in chromatin structure. And we talked about these previously, chromatin remodeling complexes, and we're going to focus on these modifications to histones, which are done by various enzymes. And these include, for example, acetylases. Acetylases tend to favor gene expression, and methylases tend to favor inactive chromatin. So looking at this process, histone modification in a bit more detail, focusing on methylation. Methylation causes formation of heterochromatin, which is a denser form of chromatin and doesn't favour gene expression. However, this is readily reversible. And here we have a diagram that illustrates simply that principle. So for example, we have here the open form of chromatin where the gene is accessible to transcription factors in RNA polymerase because it's been acetylated. We have an acetyl group attached by an acetylase. In this form, we have methylation, and as a result of methylation, we get a more compact structure, and this is not accessible, and we don't get gene expression in this format. So here's a quick review question just to see that you've got all that. With respect to interconversion between open and condensed chromatin shown below, which of the directions, one or two, would you anticipate would be the consequence of the following types of chromatin modification? So starting with A, acetylation, you go to a more open state. Methylation goes to a more condensed state. Methylation, again, condensed chromatin state. Deacetylation goes towards a condensed state, and DNA demethylation would go to a more open chromatin state. The third major category of epigenetic mechanism involves non-coding or ncRNAs, and these exert most of the effects via transcriptional regulation and may account for the great differences that we see in phenotype between species and human populations. These include microRNAs, short interfering RNAs, and long non-coding RNAs. And these participate in DNA methylation, histone modifications, and gene silencing. And once again, they have a role in cancer via alteration in gene expression, either upregulation of gene expression or downregulation of gene expression. So as we focus on the clinical significance of epigenetics, we have effects on the epigenome. As we age, the largest influence comes from the environment that we're in. So things such as diet can affect your epigenome, and we have this study here of the Dutch famine which observed changes in DNA methylation. Other stresses, smoking, stress, and changes in the fetus can occur as a result of exposure to teratogens. Teratogens are anything that affects a developing fetus. These include cigarette smoke, alcohol, and other chemicals. So you would have noticed already that we have mentioned cancer a lot, and this is one of the most studied clinical applications of epigenetics. Very first reports involved hypomethylation that was observed in cancer cells, resulting in overexpression. And a lot more studies have been done since to deepen our understanding of the role of epigenetics in carcinogenesis. What we tend to see is that methylation decreases as cells progress from a benign state to a cancerous state. We've observed hypomethylation in certain genes, like BAX2, and hypomethylation of tumor suppressor genes such as these, including BCR-ABL1, which is associated with breast cancer. However, to note, we are still in the early stages of understanding and treating cancer using this knowledge. So there's already been a number of successful clinical trials with drugs that are designed to specifically block epigenetic modifications associated with cancers. So here's a list of FDA-approved drugs that target these various processes. And we're likely to see a lot more of these drugs that come up in the next few years, blocking DNA methylation and blocking modifications to histones. So continuing with the clinical applications, we have disorders of imprinting, which results from malfunctioning epigenetic mechanisms. So we've talked a lot about how these mechanisms can cause disease, but they're also a normal part of development, where you have expression of only one parental chromosome. What must happen is we have to suppress the other parental chromosome, and this occurs normally through DNA methylation. When this goes wrong, we get disorders such as Prader-Willi syndrome, which is shown here in more detail, resulting in various observable physical defects. Here we have some other examples of diseases associated with abnormalities in gene imprinting. And we've seen some positive results from recent studies looking at treatments, and more will come out in the next few years. So the key points from today are: epigenetics refers to any factor that affects gene expression without changing the primary DNA sequence or genotype. Typical epigenetic mechanisms include DNA methylation that usually results in gene silencing, histone modifications that alter chromatin structure and therefore availability of genes for transcription, and non-coding RNA that affects gene expression. Epigenetic inheritance involves transmission of patterns of genetic expression to subsequent generations without transmitting any changes in the primary DNA sequence. And epigenetics is an area of increasing importance in human health and medical genetics because silencing of gene expression explains many disorders and phenomena such as X inactivation and genomic imprinting diseases such as Prader-Willi-Angelman and others. And of course, it has a major role in cancer. So using the concepts that we've learned about today, we can work our way through this practice question. So have an experiment with two groups of inbred mice, kept in the same environments but fed two different formulations of food. Group A gets significantly fewer calories than Group B. After one generation, they then receive an identical diet. After 2 generations, it's noted that the offspring within Group A becomes obese with an obesity-associated disease despite identical calorie content. Which mechanism best explains differences between the two groups? And in this case, the best answer is D. So we've established before that methylation, changes in methylation can occur within certain environmental stresses, and in this case, it's lack of calories. So the experiment above is a classic example of heritable DNA change due to the prenatal environment due to limited calorie availability in utero. And I mentioned earlier the Dutch famine study. There was a gene associated with that in humans, and there are equivalent genes in mice. So what you get is normally cytosine methylation of DNA leads to silencing. But when stressed in utero by limited nutrients, methylation is reduced because in part there's a limited methyl pool in order to methylate. So insulin-like growth factor 2 is required in human growth development and is maternally imprinted, and we talked about imprinting before, meaning one inherits a methylation patterning from their mother that determines expression. So when you have hypomethylation in this area, it means you get what they call biallelic expression. Both genes are expressed, paternal and maternal, allowing for growth under stress. And continued hypomethylation in this area is heritable in the offspring of those with famine, even if the next generation does not experience famine. And of course, we eliminate choice 1 because we know that epigenetics does not involve changes in the underlying DNA sequence. Practice question 2. A mother and son present to your clinic for evaluation of intellectual disability. The son, your patient, has a mild developmental delay, a long face and prominent ears. The mother reports that her brother began to have difficulties with a tremor recently. Which of the following mechanisms is most likely responsible for the cytogenic changes seen in these patients? Well, I'll give you a clue here. These facial characteristics are consistent with someone who's got something called Fragile X syndrome, as illustrated here. We haven't talked about this yet, but it is a disorder that involves DNA methylation. And so in this case, the best answer is E. So we know from before that DNA methylation is the epigenetic mechanism that brings around the longest lasting and most stable changes in gene expression, and it's the most important mechanism in Fragile X syndrome. What happens is that in Fragile X, it's a trinucleotide repeat disorder, and you get excess number of the CGG repeats in the DNA in the FMR1 gene. This is detected by the cell, and in response to this, you get DNA methylation that suppresses or silences the expression of this gene. And this increased methylation will therefore lead to fragile or about to break off experience of chromatin of the X chromosome under the microscope, hence the name. So here we have some extra resources that you might find useful. Here we have a TED talk that gives you a big picture view of chronic disease and epigenetics. We have a resource here that refers to a famous experiment with studies of rats, and we had good rat mothers and bad rat mothers. And the mothers that licked and groomed their offspring produced offspring that were less anxious and had less methylation patterns in their brains. Here we have an article from Science Direct about epigenetics, and lastly there's another review article discussing non-coding RNAs in relation to cancer.